For detailed study, please visit the website biologymom.com and follow the social media profiles. Links are in the description. Monomers of lipids. Lipids, one of the essential macromolecules of life, play crucial roles in energy storage, cell membrane structure, and signaling processes. While lipids do not have traditional monomers like proteins or carbohydrates, they are composed of smaller subunits called fatty acids. Fatty acids can be considered the building blocks or monomeric units of lipids, which is commonly known as monomers of lipids. Monomers of lipids. 1. Fatty acids. Fatty acids can be considered as the monomeric units of many lipids. These molecules consist of a long hydrocarbon chain with a carboxyl group, COOH, at one end. Fatty acids vary in length and can be saturated, no double bonds, or unsaturated, one or more double bonds. 2. Glycerol. Glycerol is a 3-carbon alcohol with a hydroxyl group, O, attached to each carbon. It acts as a backbone in the formation of triglycerides, which are a type of lipid composed of 3 fatty acid molecules esterified to a glycerol molecule. 3. Isoprene. Isoprene is a 5-carbon molecule that serves as the basic building block for several lipid classes, including terpenes, steroids, and some types of vitamins. Isoprene units can be combined in various ways to form larger and more complex lipid structures. 4. Phosphoric acid. Phospholipids, a major component of cell membranes, consist of a glycerol molecule attached to 2 fatty acids and a phosphate group. The phosphate group is further linked to various polar groups, such as choline, ethanolamine, or serine. Properties of monomers of lipids. Fatty acids. The building blocks of lipid diversity. Fatty acids are fundamental units that contribute to the structural and functional diversity of lipids. These molecules consist of a hydrocarbon chain with a carboxyl group, COOH, at one end. The hydrocarbon chain, varying in length and saturation, determines the properties and biological functions of the lipid. Saturated fatty acids, such as palmitic acid, 16 carbons, and stearic acid, 18 carbons, lack double bonds, making them solid at room temperature. In contrast, unsaturated fatty acids, like oleic acid, 18 carbons, and linoleic acid, 18 carbons with 2 double bonds, have double bonds that introduce kinks in their structure, resulting in liquid oils. Glycerol. The backbone of triglycerides. Glycerol serves as a central backbone for the formation of triglycerides, the most prevalent storage lipids in organisms. Triglycerides consist of 3 fatty acid molecules esterified to a glycerol molecule. Glycerol is a 3-carbon alcohol with a hydroxyl group, O, attached to each carbon. The esterification process involves the removal of water molecules, linking the fatty acids to the glycerol backbone through ester bonds. This arrangement allows for efficient energy storage, as triglycerides can be broken down through hydrolysis to release fatty acids, providing a readily available energy source when needed. Phospholipids. Dynamic builders of cell membranes. Phospholipids are vital components of cell membranes, providing structure, compartmentalization, and selective permeability. These lipids consist of a glycerol molecule attached to 2 fatty acids and a phosphate group. The phosphate group is further linked to various polar groups, such as choline, ethanolamine, or serine. The hydrophobic fatty acid tails orient themselves away from the watery extracellular and intracellular environments, while the hydrophilic phosphate head groups face the aqueous surroundings. This amphipathic nature allows phospholipids to form bilayers, which constitute the lipid bilayer of cell membranes. Isoprene. Versatile units of lipid diversity. Isoprene units are 5 carbon molecules that serve as the basic building blocks for several lipid classes, including terpenes, steroids, and some vitamins. These units can be combined in various ways to produce a wide range of lipid structures with diverse functions. Terpenes, derived from the combination of multiple isoprene units, are involved in various biological processes, such as the pigmentation of plants, carotenoids, and the formation of essential oils. Steroids, including cholesterol, estrogen, and testosterone, are built from the fusion of multiple isoprene units, forming a distinct structure that contributes to their hormonal functions. Isoprene-based vitamins, such as vitamin A and vitamin E, play critical roles in vision, immunity, and antioxidant defense mechanisms. Polymers of lipids. Lipids, although primarily known for their monomeric building blocks, can also form polymers under certain conditions. These polymerized lipids are less commonly discussed compared to other macromolecules like proteins or nucleic acids. In this section, we will explore some examples of polymerized lipids. Polyesterification of fatty acids. Under specific conditions, fatty acids can undergo polymerization through a process called polyesterification. Polyesterification involves the condensation reaction between the carboxyl group, COOH, of one fatty acid molecule and the hydroxyl group, O, of another fatty acid molecule. This reaction leads to the formation of ester bonds between the fatty acid units, resulting in the production of a polyester polymer. Polyesterification of fatty acids can occur naturally or through industrial processes. In nature, certain microorganisms produce polyhydroxyalkanoates, PHAs, which are polyesters synthesized from fatty acids or the derivatives. PHAs serve as storage materials and are biodegradable, making them environmentally friendly alternatives to conventional plastics. Oxidative polymerization of unsaturated fatty acids. Unsaturated fatty acids, which contain one or more double bonds in their hydrocarbon chains, can undergo oxidative polymerization when exposed to oxygen. This process occurs spontaneously under certain conditions, such as in the presence of heat, light, or catalysts. During oxidative polymerization, the double bonds in unsaturated fatty acids react with oxygen, leading to the formation of reactive radicals. These radicals can initiate chain reactions, resulting in the polymerization of multiple unsaturated fatty acid molecules. The polymerized product is often referred to as drying oils, and is commonly seen in linseed oil, tung oil, and other vegetable oils. Drying oils have important industrial applications, particularly in the production of paints, varnishes, and coatings. The polymerization process transforms the liquid oil into a solid film, providing protective and adhesive properties. Polymerization of isoprene units. Isoprene units, the building blocks of terpenes, steroids, and some vitamins, can also undergo polymerization to form polyisoprenes. Polyisoprenes are long-chain polymers consisting of repeated isoprene units joined together by strong carbon-carbon bonds. One notable example of polymerized isoprene units is natural rubber, which is a polyisoprene polymer produced by various plants. Natural rubber possesses excellent elasticity, making it valuable for numerous applications, including tire manufacturing, industrial products, and consumer goods. Synthetic rubber, such as styrene-butadiene rubber, SBR, and polyisoprene rubber, IR, is also derived from the polymerization of isoprene units. These synthetic rubbers exhibit properties that make them suitable for diverse industrial applications, including automotive components, adhesives, and seals. For detailed study, please visit the website biologymem.com, and to remain updated, follow all the social media profiles. All the links are in the description.